Good morning. Happy Wednesday. Happy Wednesday. We are looking at the study guide worksheet today. And making sure that everything, because we're testing tomorrow. Testing for you all this Friday. Testing. So we're going to make sure all our worksheets are in. I'm going to go over the study guide. So if you haven't pulled that up or downloaded that, make sure you do that. All right, it's doing that thing again where I still see you in my waiting room, but you're already here. Okay. One new person. Mm-hmm. Okay. Also, while we're waiting, so what we're going to start doing next week at the beginning of class is to look at in progress learning, the questions that you all struggle with. And I'm going to look mostly at the ones that of topics that we've covered, but we'll also look again at I'll say the other ones that for topics that we didn't cover until we actually get to them. Okay. So we are making sure all worksheets are submitted. I love it that everybody's here. So I can do attendance real quick. And then we're going to look at that worksheet. So I'll give you a few minutes if there's something that you have outstanding, work on that, and then also pull up the study guide at the end of the module so that we can go over that. Okay. Appreciate y'all being punctual on time. Showing up every day makes taking attendance so much easier. Alrighty. That's not the one. Alrighty. Let me see if I can. All right, I'm trying to pull up the meiosis worksheet at the same time. All right, so what I want you to do is to try to have both your mitosis and your meiosis worksheets handy so that you can look at them while you're trying to put the little X's in all of those columns. Okay, so I'm trying to have several pages open at the same time so that we can jump back and forth. See, this is gonna work. What in the world? Okay. All right, so another thing that you can do while you're doing that worksheet, in the unit that says meiosis lesson. So if you're in Canvas and you scroll down, you have mitosis and meiosis side by side. Okay, so you can be able to look at the worksheet and kind of hop back and forth. All right, so let me see if I can do that. All right, so if you look at this picture of these two side by side, then you can, where's my worksheet? Oh, yep, yep, there we go. All right, so then you can uh, compare one to the other side by side, produces 2 daughter cells, produces 4 daughter cells. So if we jump back over here to that one, and we look at what it produces at the end of the day, this one shows you that it produces 2 daughter cells. This one, meiosis, shows you 4. It may not look like 4 cells, but it should be. Actually, if you're looking at this right here, those are the 4 cells, the gametes right there. This right down here is what happens if you combine one gamete with another gamete, you get a zygote. So the only time you get a zygote is if a zygote is a fertilized egg cell. So the egg and the sperm will move on to unite and form a fertilized cell or a zygote. So again, know your vocabulary. The gametes, so this one is showing 4 gametes. One of them is turned in. If this was a female, you would have 4 circles. If this is a male, you'd have 4 sperm cells. Okay, but you'd have 4 of them. Okay, and you take 1 sperm and 1 egg and you combine it to make a zygote. All right, now these are all going to be slightly different than the original cell. Okay. All right, notice this one has a blue one in it. There was no blue in it when you started. So there's some variation when you end up with a zygote, as opposed to these two on mitosis look exactly like the ones you started with. Okay, so that's another point on the graph. All right, so we've answered those two. So 2 daughter cells is mitosis. You should have an X there. 4 daughter cells, you should have an X on meiosis. Produces daughter cells that are exact copies of the parent. That's mitosis. I just showed you that. Produces daughter cells that are not identical, and that's meiosis. And again, I just showed you that. Looking back at the diagram, because we had some variation. All right. Has 2 divisions. So meiosis has a meiosis 1 and meiosis 2. So that's what so their X should go there. Has 1 division, and that would be mitosis. Okay. The next one occurs in sex cells. That would be meiosis. Mitosis is all your other cells. Okay, all your other body cells. Somatic cells, again, are your other body cells. So occurs in sex cells is meiosis, occurs in somatic cells is mitosis. Somatic, again, is a vocabulary word for all the other cells, non-gamete cells. Okay. Allows for genetic variety. That's the word I used earlier was variation. So variation is going to be meiosis. Produces haploid cells is also going to be meiosis. If we come back over here, let's see, where do I want to be? There. We come back over here again. We need to remember that mitosis has diploid, and then you're going to separate those when you get down here into haploid cells. So it may look like it starts with diploid, but it's going to create haploid cells. So the first part of meiosis looks like mitosis, and then those resulting cells split again. So here we get 2 cells, and then those 2 cells are going to make 4 cells, because each of those 2 are going to split again. And they're going to create haploid cells until fertilization takes place. All right, moving right along. Everybody with me? Anybody need me to repeat something? Hopefully all of you are following and not trying to work on makeup work while I'm giving you the answers to the study guide. All right, so here we are, haploid cells. Homologous pairs, mean the pairs are the same. Homologous means the same. So the chromosomes in mitosis, and actually both of them have homologous pairs, so you can have an X in both. But by the time we get to meiosis 2, we don't have homologous pairs anymore. All right, involves replication of chromosomes. So then that happens in both cases. It starts off with replication and then the replicas split. So that's in both cases. Crossing over only occurs in meiosis, and crossing over, and we need to come back over here and look at that word, crossing over, crossing over. What is crossing over? And I guess that's something she tells you in the video because they don't seem to have. Okay, let's see. Crossing over is number 5. Let's see.
You're excited, aren't you? Because there are 4 stages in mitosis that we're going to look at. And you're excited, aren't you? Because meiosis 2 is essentially mitosis with half the chromosomes. So the reduction division has happened. We now, if I were to draw a picture of what we have at this stage in the game, you can imagine that we've got one big chromosome.
Okay, we're not going to watch the whole thing.
Straightforward, isn't it? Okay, we're going to, why? Why do we do this? There are some parts of meiosis. Number 1, it gets us a haploid chromosome number. But number 2, there are things that are happening in meiosis that promote genetic diversity. So we're going to look at those things. For 1, we have the process of crossing over. Let's look at that more closely. For 2, and you can actually see that, her, we actually have a process called independent assortment that is significant for producing genetic diversity. And then if you start thinking about, whoa, how many possible sperm are there and how many possible eggs are there? And then how many possible babies can you make? Holy random fertilization madness of fun times and diversity.
That's why.
I'll be right back, and let's talk about crossing over.
And they don't have the one that talks about crossing over. So I tell you what, let's take a break here and let me, she skips right over it. And they ask you, that's a question on the test. What is crossing over? So let me help you with that. An independent assortment. And some of these are way too long. Okay, I got one that is kind of short, so let's look at that. All right, so this is a short one that explains what is meant by synapsis and crossing over.
During meiosis, synapsis and crossing over occur between two chromosomes in a homologous pair, and from an evolutionary standpoint, by exchanging genetic information between the mother and father chromosome, there's an advantage of increasing variability within the gametes produced from meiotic division. Synapsis refers to the connecting of homologous chromosomes together, and this connected pair forms what we call a tetrad. Tetra meaning four to refer to the four sister chromatids present, 1, 2, 3, and 4. In crossing over is the actual exchange of genetic information, so this is crossing over, where pieces of both chromosomes break off and then reattach to the opposing homologous chromosome. So the completed exchange creates recombinant chromosomes with 4 different sister chromatids, as opposed to only 2 different types of sister chromatids before the exchange. Remember, these 4 sister chromatids are then separated into 4 different gametes through meiosis 1 and meiosis 2. Interestingly, not all genes are equally likely to cross over to another chromosome. Depending on the distance between 2 genes on a chromosome, there will be a different probability of these genes separating.
All right, I'm going to stop here for a minute. All right. And it's going to be important when we do the next lesson on genetics, because we're actually going to look at mapping of chromosomes. So when they say homologous, you'll understand it better when you see a whole thing. They're going to be the same size, and the same information that's on this blue one is on the green one, except it might be slightly different. So while we might have hair and eye color on this blue one, it could be brown and blue, brown hair, blue eyes, whereas we still have hair and eye color on the homologous pair, but it might be blonde and brown eyes. Okay, so it's coding. When we say homologous, it's coding for the same characteristic. That's what we mean by homologous. Okay, over here on the end, notice they kind of do a little twisty dance, and then a piece of the green one is now on the blue one, and a piece of the blue one is now on the green one. So that is the crossing over part. So when these split up into 4, you're going to have one that's totally blue. You're going to have this one.
Makes more sense when you look at a simple.
Hold up. When they break apart during meiosis, this whole blue one will go one way. This whole green and blue one will go one way. This blue and green one will go one way, and this totally green one. So that's 4 different chromosomes that will end up in the 4 resulting cells in meiosis. So, okay, so the one that gets fertilized, you don't know which one is going to get fertilized. It could be the green and blue one. It could be the blue and green one. It could be the total blue one. It could be the total green one. That's where we get the variation. You don't know which one of these is going to get fertilized. All right, let it finish. Oops. Okay, stop. Close that. Continue. See, that thing is in the
simple diagram like this one. And so if you see, crossing over between A and B is less likely to occur than crossing over between A and E, because there's only this small amount of space to cross over between A and B, whereas there's this much space to cross over between A and E. So a crossing over between A and E could happen anywhere between all this space. So it could happen here, it could happen here, it could happen here, it could happen here. There are a lot more different possibilities than between A and B. So you can see that the rule is the further apart 2 genes are, the more likely a crossover will occur and the genes will be separated.
Okay, so I just wanted to give you a quick look at what is crossing over.
Meiosis is divided into 2 parts, meiosis 1 and
I'll leave
meiosis 2, which are again divided into 4 more parts: prophase, metaphase, anaphase, and telophase. You should recognize that these are the same names for the mitosis phases, but they accomplish
All right,
different
remember
things.
PMAT, prophase, metaphase, anaphase, telophase, PMAT.
Meiosis I is also called reduction division. Before it occurs, a cell will undergo the same process of DNA replication as in mitosis. So, when the cells begin the meiotic cell division process, there are 2 sister chromatids per chromosome. Unlike mitosis, however, the sister chromatids do not separate in meiosis I. Instead, the homologous chromosomes separate. The homologous chromosomes are the 2 chromosomes that code for the same gene in a diploid cell. Remember, we said that humans, in each pair of homologous chromosomes, one comes from the mom and one comes from the dad. And so, for example, if we were talking about eye color, both these chromosomes will have the genes for eye color, but let's say the mom chromosome will have the genes for green eyes, whereas in the dad's chromosome, the dad will have the genes for brown eyes. So this is the difference between homologous chromosomes. One comes from the mom, one comes from the dad, but they code for different variations on a specific trait. And so when these separate, the cells go from a diploid count to a haploid count. Synapsis and crossing over also occur in meiosis I, which is the process of exchanging genetic information between 2 chromosomes in a homologous pair. And so these 2 processes are discussed in more detail later. Once the exchange is complete, the homologous chromosomes separate by lining up randomly at the center of the cell and moving to opposite sides, and the resulting haploid cells are made genetically unique. And so in meiosis II divides the cells a second time to separate the sister chromatids in each chromosome. So it's very similar to mitosis in that the chromosomes line up and are separated along their centromeres. And this division maintains the haploid count in cells. However, this stage is different from mitosis in that it does not create genetically identical haploid cells. Because of the exchange of genetic information in meiosis I, the sister chromatids in each chromosome are already different.
I'm going to pause right there because it'll probably run out before I can point this out. All right, so if you're looking at this part right here is meiosis I, and the first cell is showing that they replicate. So you had these single ones and then they ended up being double. Okay, but the copy stays together. Then what happens in the third one, those copies start to move apart. And by the time you get to B, you're getting one set of each of these, one short one, one long one, is going to separate. So they're kind of like the line is going to go,
Thus,
line is going to go down the middle so that you get one short one and one long one over here in C, and one short one and one long one over here. Then those cells are going to go through a division, but they're not going to replicate. So those pairs that are being held together by the centromere are going to pull apart. And when the cell divides into, each one of those is going into a separate cell. So we're creating 4 cells from the 2 that were created from meiosis I. During meiosis II, we create 4 cells with one N. You see up here at the top, 1 N. We're going from 2 N to 1 N, where they were duplicated and now they're singles. And notice when she said the variation, this one up at the top is kind of red and blue. This one down here at the bottom is red and blue. So you've got a little variation at the top of this one, at the bottom of this one. The short one also ended up doing crossover, so they have some variation there. So each of these cells is different None of them look exactly like this first one that we started with in A. Okay, so there's variation there. All right, and I'm going to stop this right here and come out of it, and we're going to go back to the worksheet. Okay, all righty. So that, where were we? We did variation, haploid cells was meiosis, pairing of homologous occurs in both, involves replication, occurs in both, crossing over only happens in meiosis, produces gametes only in meiosis, associated with growth of asexual reproduction and growth and repair is mitosis. That's all the other cells, your somatic cells, they do that. Sexual reproduction is going to be meiosis. That's the only thing meiosis is for, producing sex cells. Chromosome number is maintained in mitosis. It is reduced in meiosis. Okay, so that's half. And spindle fibers are required in both cases. Okay. Like I said, you don't have to do the Venn diagram, but the Venn diagram simply wants you to take all of this information up here. The things that have both will go in the middle. The things that are only mitosis would go on this side. The things that are only meiosis will go on that side. All right, moving right along. All right, sexual reproduction requires two parents. Asexual, one parent. Methods of, they are both methods of reproduction, so you would have an X in both columns. Creates more genetic variety would be sexual reproduction. Asexual is like mitosis. It only creates exactly what you started with. Okay. Short lifespan would be sexual reproduction. Asexual reproduction. Copies the DNA, both of them will copy DNA. Crossing over only happens in sexual reproduction. Asexual is quick. More complex organisms is sexual. In other words, simple organisms usually do one of the variations of asexual reproduction that we had on our worksheet. It takes a long time for sexual reproduction. Autotrophs are things that feed themselves, so those are plants. So we can have asexual reproduction in autotrophs, but heterotrophs, and then require sexual reproduction, but heterotrophs can also be like cuttings, where we can take a plant that is a heterotroph and create another one by doing cuttings. Okay, so that's what that one is. All righty. And our cancer one, I'm trying to get through before the bell rings. All right. Mutations cause growth. That's cancer. Normal cells are normal. Grows uncontrollably. That's cancer. Cells divide. Both of them, you would have an X in both columns because all cells divide during normal cells and cancer cells. Cancer is damaged DNA, and both undergo mitosis. Okay, so again, I know I did that fast. So there's an X for mutation under cancer, not normal. Grows uncontrollably, cancer, not normal. Cells divide, both of them. Damaged DNA is cancer, and mitosis is both. Okay, all right. Please, please, please look at your oh, I thought I had that up somewhere. I don't have it open twice now. But please make sure that you go through this. Many of you were confused by this. I'll tell you what the answers are now. Most of you were good at putting the correct phrases underneath because all you had to do was copy what it said for that, but you were not good at associating the pictures. So diploid cell was F, and that's this one. So that means you have two sets of chromosomes going on here. All right, prophase 1 was G, and that's this one. You can, Andre, you can resubmit. Metaphase is D. Anaphase is H, and I'm going through this quickly because the bell's about to ring. I say bell ring, the time is about to be up. Telophase is J. Prophase is A. And this is where y'all started to get them wrong. Metaphase 2 is B. Anaphase 2 is E. Telophase 2 is I, and the gametes are C. I think most of you realize that these 4 cells were the gametes. So that one, most of you always got right. But all the ones in between that you had trouble with. Okay. All right. So mitosis, meiosis, asexual reproduction, and cancer. Those are the topics for the test. For you, it's on Friday. Please study, study, study. Please also have all missing work in by Friday. Progress reports are posted on Friday. You don't want to have any missing grades reflected on the progress report. Okay. All righty. All right, Raymond, I do see what you put in the chat. That is something I need to open. Okay, it won't let me. So I think what I need to do, Raymond, is just open that assignment again and hold on. Mitosis interactive. It might be closed. Let me check. Yes, it's closed. So I'm going to open it. And then if you would please put it in that folder, because I can't open what you just sent me, but you can upload it in the file as soon as I change the date to Friday. All right, so for those of you who hadn't done your mitosis interactive, that column is now open until Friday. And so you can now submit that without it being late. Well, I will say late, but okay. Any questions before you go to lunch? I see somebody's already left. A couple of people just said, bye, it's lunchtime. Have a good one. See you Friday.